Your body is constantly tearing down its own proteins. Every second, muscle fibers and cellular structures are dismantled, releasing their building blocks, amino acids, into your system. But hidden inside every one of these amino acids is an atom of nitrogen. While we can easily pack away extra fat and carbohydrates for later, human biology never developed a way to store free amino acids. They have to be processed immediately. This diagram outlines the body's mandatory disposal route. We start with the raw amino acids in blue. The system strips away their nitrogen to form ammonia, shown in red. That ammonia is then quickly converted into a safe molecule called urea, highlighted in green, which travels to the kidneys for excretion. That red ammonia stage is the danger zone. Keeping that toxic compound contained during transport requires a highly precise, microscopic chain of custody. The first step in this logistics network is transamination. Instead of just ripping the nitrogen off and dumping it into the bloodstream, the body carefully hands it from one molecule to another. Here, an amino transfer enzyme executes the transfer. It lifts the nitrogen-containing amino group off the original amino acid and attaches it to alpha-ketoglutarate. This creates glutamate, acting as a secure biological shipping container. It locks the nitrogen down for safe transport through the body. In tissues like the liver and muscle, specific enzymes handle this cargo loading. This schematic shows the two primary handlers, ALT and AST, funneling nitrogen from various amino acids straight into glutamate. Because of this constant shuttling, toxic nitrogen is never left to float freely through your veins. It stays safely packaged for its journey to the liver. Once the glutamate reaches the liver, it drops off its cargo. Here, a process called oxidative deamination finally cuts the nitrogen group away from its carrier molecule. As you can see in this animation, the blue amino group is cleaved off and instantly becomes free ammonia, that glowing red NH3 icon. This is a highly volatile and dangerous compound. If that ammonia escapes the liver, it penetrates the blood-brain barrier. Once inside the brain, it triggers the astrocytes, vital support cells for your neurons, to absorb water and swell rapidly. This puts the liver on a ticking clock. It has to neutralize every molecule of free ammonia the exact second it's generated, before it can breach the central nervous system. To execute this rapid neutralization, the liver fires up a specialized metabolic engine, the Urea Cycle. Looking at this cycle map, deep inside the mitochondria, free ammonia bonds to carbon dioxide, forming carbamoyl phosphate. Moving down, aspartate donates a second nitrogen atom. Finally, in the top right, arginine is cleaved, safely releasing urea. The liver burns a massive amount of cellular energy to keep this cycle turning. But that cost is strictly necessary. It converts a lethal neurotoxin into a harmless, water-soluble waste product that the kidneys can easily filter out. But what happens when a genetic defect or severe liver disease shuts this biological machinery down? The result is a condition called hyperammonemia. The urea cycle stalls, and toxic ammonia backs up, overflowing from the liver directly into the systemic circulation. The clinical progression is rapid and devastating. Patients first exhibit severe lethargy and vomiting, which escalates to cerebral edema, coma, and without immediate intervention, death. This system map illustrates the body's three-step rescue operation, constantly running in the background. First, transamination collects the waste. Second, deamination releases it inside the liver. And third, the urea cycle neutralizes it. Human cognition and life itself depends entirely on this microscopic waste management pipeline operating continuously, without a single flaw.